Thank you for inviting me today to talk about a little bit of our research at Axolabs. And our field is oligonucleotide field, and over the last years, we haven't seen progress in this field with the approval of antisense drug and also with reports of promising results in the clinical trials. Despite all these advances, the key problem is still the safe and effective delivery to the respective target organs. The LNPs are a clinic advanced technology. We see also today that delivered to other organs other than liver and tumor has been reported, but the efficacy and the safety profile requires further improvements. In opposite to further promoting the oligonucleotide delivery in the different cell types, our strategy focuses on understanding the LNP modes of action and the biology. And to address all these questions, we work very closely in the Axolabs with the different teams, from the lead identification to the manufacturing of the RNAs, to the biology, of course, the delivery, and the bioanalytics group. Let me use the liver to explain our strategy. The liver, as is in the scheme, is composed by different cell types. We have hepatocytes, we have stellate cells, endothelial cells, and Kupffer cells. To monitor the cell tropism of the formulations, we need to measure the knockdown on the specific cell type. And for that, we need to identify different cell markers. And for the hepatocytes, we've identified factor 7 for hepatic stellate cells, reelin siRNAs, endothelial cells, CD31 and TEC, and for the Kupffer cells, CLEC4F. Depending on the target cell, we need to find an appropriate formulation that then can be generated to knock down only on this cell type. Here we explain our way to identify the RNAs. First, the bioinformatics group using in silico design identify a high number of siRNAs to be tested in vivo. In vitro. This silico design is a proprietary algorithms that with high off-target predictions. From the identified leads, we go into the small scale and we synthesize the different RNAs. They are tested in vitro. We check the dose response, the cytokines induction, and the stability of the lead candidates. We can do optimization of the different lead candidates. We verify the in vivo of targets, and then we go to verify the in vivo activity. We also synthesize and develop lipids for the formulations. Here we have examples of our proprietary lipids. These lipids can be synthesized by multi-synthesis or just one synthesis reaction. The most important part is really the purification of these cationic lipids. As we can see in the top HPLC trace, we have always very dirty compounds, and we always try to purify in order to have higher than 95% purity of the lipids to go in then in the next phase. Here we show the approach that we do for our LNPs. We first have our initial formulation where we just look to the different lipids. We formulate them, of course, with our nanoassembler, And then we characterize the particles in the different aspects. We do an in vivo testing, and all the active candidates go in a second round where we do optimization with fine-tuning of the composition. We look again to the particle characterization. We see the reproducibility of the manufacturing and the long-term stability. We then look to in vivo testing with the different mRNA knockdowns, safety profiles, and our goal, the cell type tropism. Here I show the different characterization of the formulations. In the top, we have the table with the different lipids in the same formulation. We can see that we have a very different range of sizes. It goes from 60 nanometers to 160. The zeta potentials are also very different, from near neutral to very negative particles. The encapsulation is also different depending on the formulations. If we look to the two pictures, the XL formulation and the XL8 formulation, if you look to the table, have the same size, but they have very different structures. The top one have multiple vesicles, and the top or the down picture, we can see they are solid nanoparticles. When we formulate different siRNAs with the same formulation, we can see they have very constant parameters. We have more or less the same size, the same zeta potentials in the same encapsulation. So NanoAssembler is really a great instrument. We can go from one formulation to another without much variation. Here we show how we assess efficacy and the safety in mice. We have different routes of the administration of the drug. Normally with the LNPs is intra-IV. We assess in day X our efficacy. Not only the efficacy in mRNA, but also in protein level. We also collect organs, and we look to the clinical chemistry, hematology, and organ pathology. In the meantime, we can also do intermediate blood collection, where we can look for other parameters like cytokine induction. Here we have the results for 3 different formulations. They were formulated with all the different cell markers for the different cell types. They were injected by IV, and for 8 hours after injection, we measured the target mRNA in the liver. We have for the different colors the different lipids, and for each we did 3 different doses. We have, for the hepatocytes, is a very clear dose response curve, with XL10 being the best lipid at very low doses. For stellate cells, we can see just the XL8, the compound in red, as dose response curve, and is the only one that goes to the stellate cells. For Kupffer cells, we can see that at very high concentrations, XL8 and XL10 knock down the cell marker. And the XL8 we can see over the range of the other administrations doses that is saturated. XL7 we don't see a knockdown, and we can see that there's also an increase of the blue curves compared with the controls, and that is normally something that we see with RNAs when we don't have so much knockdown. And also Kupffer cells are an immune cell line, and that can also already translate that there's some toxicity involved with this formulation. In the endothelial cells, we can see only at very high doses XL8 40% knockdown of CD31. So we can see for the different markers, we can have different lipids that knock down specific cells. We did the same experiment using GFP transgenic mice, and we formulate the formulations with GFP siRNA, and we assess the liver 4 days after. The GFP was analyzed by confocal microscopy, and the cell types were counterstaining with the respective antibodies. In the top, we can see hepatocytes. We have in the left a PBS control, and in the right, the LNP formulation. We can see the reducing of the GFP. For the Kupffer cells, we can see in the top the red cells that is marked then in the bottom with the different arrows, and we can see the reducing of the green fluorescence in these cells, and not in the hepatocytes that are the bigger ones. In the endothelial cells, we also have the counterstaining with CD31 in red, and in the formulation we can see the reduction of the green. Is not totally removed the GFP, but we can see reduction on the GFP level. We also don't want only to measure siRNA in the tissues. We would like also to know what happens to the lipid. And for that, we develop a method to extract the tissues and the lipid from the tissues and analyze the different quantities in the different organs. So here we have XL8 from the experiments before, and we have the different dosage groups. We can see that in the liver we can find the biggest amount of the lipid, and depending on the dose we can see if very scalable is 5 times less what we expected by the injection. In spleen, we can still measure the lipid in kidney in higher doses, and in the lung in very high dose. So like that, we can have only not a pathway just for measuring the lipids and the siRNA, and follow what can happen to the lipid during the in vivo experiment. We are not only interested in liver, we are also interested in, of course, other organs. And the same is true for lung. Lung is a much complex organ, as we see here. They are different ways to administrate the drugs. We have the direct and the systemic way. And we have various cell types in the upper airways and in the lower airways. We were able to identify 8 different cell types siRNAs, and we also have a direct assay for the mRNA knockdown as a consequence of the cell type delivery. Here I show you some results from a collaboration with Engine. We formulated the RNAs, endothelial cell markers RNAs with the Engine formulation. And we introduced IV 2 mg per kg, and 48 hours we measure the mRNA knockdown. In this case, we have in the left CD31 mRNA silencing. We have 2 different sequences, and we can see that we have around 80% knockdown with CD31. The same is true with the TEC or Taiwan 2 mRNA silencing. We were confirming that a second endothelial marker, it was able to be knocked down in the endothelial cells using this formulation. With this, I hope that I show you that we can have a very high throughput screening of the RNAs. We also show that the cell type specific targets is an important screen tool for development of the LNPs as delivery systems to other cells and organs. The different LNPs can be selected and further engineered to target the preferential and desirable cell type. And I hope that I could show that we have a streamlined process from the lead identification to the preclinical studies. And with this, I thank you very much for your attention.
Cell specificity profiles of the different lipids, is that due to opsonization with specific proteins, or is it physical?
So they have different mechanisms of entering in the cell. We know that a few of them enter for ApoE-mediated and others not. The differences so far we were not able still to figure out is just because of the mediation, or is because of the different parameters of the formulations, the potential size, etc.
Okay. Thanks a lot.
I have a question here from one of our followers from the webcast, Alexander Zaifalian. Looks more like a general question. Has any regulatory bodies such as the FDA or EU published what toxicology and biocompatibility tests are required before going to clinical trials?
I know there is a regulation, but I'm not sure now. Do you know, Monica? I know that we need to follow the FDA rules and there's safety profiles, but I cannot say by the top of my head.
Just a quick question. Probably I missed something. What is really the aim of this lipid quantification, and what kind of lipid do you really quantify by LCMS?
So what we want to see is what happened to the lipid in vivo, because it's one of the questions, stays there forever, goes away, is toxic in some way, and where is going, how it distributes. We can follow that very well with our mRNAs, RNAs, But so far, we never had a deep look what's happening to the cationic lipid, especially. And this is the goal, to have a deep look where the lipid is localized, and if also gets in connection with the different activities. If we have too much in the lung, do we have also knockdown in the lung with these formulations?
Thank you.
Do you take your encapsulation efficiencies into account when you're dosing?
No.